Okay, Rajakumari, now you can start your presentation.
Okay, good evening, everyone. Today I'll be giving a presentation on my case study title, which is about in silico docking studies and ADME prediction of inhibitors of NF-kappa B as a promising therapeutics for Alzheimer's disease. So generally, this will be a general outline of what I'm going to be presenting, where first I will give an introduction on NF-kappa B, followed by the NF-kappa B family, the mechanisms that are involved in NF-kappa B activation pathway, then focusing mainly on the molecules that I've used for the docking studies, which is endographolide and 12-13 dihydroendographolide, which is HAGP and its benzylidine derivatives, and then focusing on the objective, the methodology, as well as the results and discussion. In the results and discussion, basically, I'll be presenting the results that I've obtained from the docking studies, calculation of prime MMGBSD descriptors, as well as prediction of ADME properties. Then finally is the conclusion. So generally, NF-kappa B is basically a transcription factor that is found commonly in the neurons and glial cells. And this NF-kappa B is known to play an important role in various kind of conditions such as in inflammation, immune response, apoptosis, as well as cellular response. And improper regulation of NF-kappa B has been linked to development of various kind of conditions such as cancer, inflammation, autoimmune disease, as well as Alzheimer's disease. So this is basically the NF-kappa B family, whereby they consist of 5 polypeptide subunits, and these 5 polypeptide subunits are the P50, P52, P65, or known as the REL-A, REL-B, as well as C-REL. And generally, the NF-kappa B is known to exist in the form of dimer, whether homodimer or heterodimer. And the most common form of this NF-kappa B is in the form of heterodimer, P50, P65 subunit. And even in the brain, commonly the P50, P65 heterodimer are the most common. And this REL homology region is basically involved in 4 important functions, that is for dimer formation and for nuclear localization, for DNA binding, as well as for inhibitor kappa B binding. And the REL homology region can be basically divided into 3 structural regions, which is the N-terminal domain, the dimerization domain, as well as the nuclear localization signals. And as we can see from this diagram, basically besides then the NF-kappa B family, there is also presence of inhibitor kappa B family. And the function of this inhibitor kappa B is mainly to make the NF-kappa B to remain in inactive form in the cytoplasm. And inhibitor kappa B family, basically they have various subunits such as IKB alpha and IKB beta. And what can be seen the difference between inhibitor kappa B family and the NF-kappa B family is basically in the inhibitor kappa B family, there's the presence of ankyrin repeats, which is highlighted in blue color, and their absence in those of NF-kappa B family. So this is the mechanism of NF-kappa B activation pathway, whereby they have 2 main pathways, the canonical pathway and the non-canonical pathway. And since most of the conditions such as cancer or inflammation actually is linked with canonical pathway, so in this presentation I'll be generally focusing on canonical pathway. So basically, once the molecules such as TNF alpha or interleukin 1 binds to the respective target, they will then cause us into activation of this IKK. It is basically known as inhibitor kappa B kinase complex, where this complex exists in 3 subunits, the alpha, beta, as well as NEMO. NEMO is the IKK gamma form. And from these 3 subunits, the IKK beta is the one that is responsible for its function, that is to cause phosphorylation of the inhibitor kappa B alpha. As I've stated earlier, that NF-kappa B, most of the time they remains inactive in the cytoplasm because of the presence of inhibitor kappa B alpha. So once this IKKB causes phosphorylation of the IKB alpha, then it will leads into ubiquitination and further causes degradation of the IKB alpha by the proteasomes. So this eventually leaves the P50, P65 heterodimer in the active form. And once they are in the active form, they are able to translocate into the nucleus and causes further, once it enters into the nucleus, then they can actually cause into various kind of conditions, such as they can cause into a development of cancer, inflammation, and other form of diseases. So basically, for the docking studies, I have actually worked on 66 different molecules, where the first set I've actually used endographolide and its benzylidine derivatives. Why basically endographolide is selected? Mainly because it is known to have a good anti-inflammatory property. And AGP and the benzylidine derivatives has also been tested for anti-cancer activity. So now we want to see whether this molecule actually acts as a potential inhibitor for NF-kappa B or not. So as we can see that basically for AGP and the benzylidine derivatives of AGP, there is similarities in the scaffold, which is highlighted in green color. And the differences is that for AGP, there is a presence of two hydroxyl group at carbon number 13 and 19. And sorry, carbon number 3 and 19. While for the benzylidine derivatives of AGP, basically there is a presence of benzene ring attached with various kind of substituents. So the type of substituents that I've used for docking studies is shown in the table below. 21 types of derivatives of benzylidine derivatives of AGP. Next is the 12-13 dihydroendographolide HAGP and its benzylidine derivatives. This is basically a new set of molecules which not been synthesized yet. And what is being known is that basically this HAGP, they can exist in two configurations, mainly because there is an absence of double bond at carbon number 12 and 13. So there is a presence of configuration, they can exist in R form and S form. So for these docking studies, basically I have docked 22 molecules of R configuration and 22 molecules of S configuration. So what we can see is that for HAGP and benzylidine derivatives of HAGP, they share the same backbone of the scaffold, which is highlighted in blue color. And the differences can be seen is for HAGP, there is presence of two hydroxyl group at carbon number 3 and 19, while the benzylidine derivatives of HAGP, basically there is presence of benzene ring attached with various kind of substituent. And the substituents that I have used, the 44 different types of molecules that I've used for docking is as shown in the table below. So basically the objective of the study is mainly to perform docking studies for AGP, HAGP and its benzylidine derivatives, and followed by calculating the free energy of binding, that is basically to identify which molecules forms the most stable interaction with the target, where in this case I've actually used DNA binding site of P65 as my target. And finally is to predict the ADME properties of the 66 molecules that's been docked in order to identify if they show any drug likeness like property. So moving on, this is the methodology, whereby the first one is on protein preparation. So the protein here is the target that's been selected, which is the X-ray crystal structure of NF-kappa B P65 homodimer complex with DNA target, where the PDB entry is 2 RAM. And once the protein has been selected, it was refined and prepared using Schrödinger protein preparation wizard tool. Next, ligand preparation. Basically, all the 66 molecules were sketched using Mastro software, and they were prepared using LeakPrep. And the ligand minimization was done using molecular mechanics force field without default settings. And for the docking studies is where all the prepared ligands and the protein were docked using Glide XP docking program. And for calculation of prime MMGBSA is where basically the post-viewer file, that is the file that exists after the docking was done, was used to set up the calculation. And prediction of ADME properties was done using KeyProp module of Schrödinger. So basically, this is the results section, where a results and discussion part, where the first part I will be discussing on the target that I've selected, which is the DNA binding site of P65 subunit. And there is basically presence of 5 key amino acid residues, which is shown in those highlighted with yellow color. That is the arginine 33, arginine 35, glutamic acid 39, tyrosine 36, as well as arginine 187. And this is actually showing the position of these 5 key amino acid residues in the DNA binding site of P65. So moving on is, I'll be focusing first on the AGP and its benzylidine derivatives. And this is the image of the 3D superimposed image of the molecules that has been docked with the target. And it has been found that basically for almost all of the molecules, they were found to bind at the binding pocket of the target, which is the DNA binding site of P65. And we can see here basically the molecules were found to bind with the glutamine 39, tyrosine 36, arginine 187, and arginine 33, which is the key amino acid residues that have been shown previously. And from the 22 molecules of AGP and its benzylidine derivatives, basically I've selected one example, that is AGP11, and the structure of the AGP11 is as shown in the those highlighted in purple color. And what we can see is here on those highlighted with orange color is where how the AGP11 binds with the target. And we can see basically there is a presence of hydrogen bond interaction. That is where the hydrogen atom of the OH group of the lactone ring are known to form a significant hydrogen bond interaction with the oxygen atom of the OH group of phenyl ring tyrosine 36 at 2.17 Armstrong. And there is also presence of pi-pi stacking, which has been highlighted in the red color, where we can see there is basically the phenyl ring of AGP11 forms a very strong pi-pi stacking interaction with the phenyl ring of tyrosine 36. So basically, this is the graph that shows the interactions of all the study molecules with the crucial amino acid residues of DNA binding site of P65 target. So the first graph here is actually showing on Coulombic interaction for the 22 molecules of AGP and its benzylidine derivatives. So we can see from here is that AGP07 is known to have a significant Coulombic interaction with arginine 187, and AGP04 is known to have a moderate Coulombic interaction with tyrosine 36, and there is also a moderate form of interaction shown by AGP13 with arginine 187, and AGP09 is known to show a weak Coulombic interaction with glutamic acid 39. So the second aspect will be on van der Waals interaction. Using the similar molecules, it has been identified that AGP16 is known to show a significant van der Waals interaction with tyrosine 36. And AGP0304 is also known to show a moderate van der Waals interaction with tyrosine 36. And we can also see that for almost all molecules, they form a moderate van der Waals interaction with arginine 187. And this is the graph that shows the hydrogen bond interaction, whereby we can see that for almost few of the molecules, tyrosine 36 plays an important role for hydrogen bonding interaction, where we can see that for AGP04, 10 as well as 15, they form a significant hydrogen bond interaction with tyrosine 36, while those AGP06 are one of the moderate hydrogen bond interaction shown with tyrosine 36. So this graph is actually showing the calculation of ligand-free energy of binding with the DNA binding site of P65 target using the MMGV assay approach. And what it can be seen that all molecules of AGP and its benzylidine derivatives anchored well inside the target of DNA binding site of P65. In particular, we can see that AGP05, AGP11, AGP14, and AGP16 forms a strong interaction with the DNA binding site of P65 target. Next, this is the ADME properties for AGP and its benzylidine derivatives. I've selected a few parameters to see the drug likeness property shown by these molecules. And what it can be seen that for these 22 molecules, basically they show a good drug likeness like property, whereby they falls within the acceptable range for each of the parameter. And this is basically the second set of molecules that I have docked, that is the HEGP and its benzylidine derivatives, that is for both R and S configurations. As we can see in the figure 10, is showing the 3D superimposed image of the R configuration of HEGP, and the figure 11 is showing those of S configuration. So one clear difference that we can see from here is that for the R configuration, most of the HEGP and its benzylidine derivatives were known to bind to the binding pocket of the DNA binding site of P65, where we can see that they tend to bind with those arginine 187 and tyrosine 36. But when we see for the S configuration, most of the HEGP and its benzylidine derivatives were found to bind outside of the binding pocket of the target, meaning that it doesn't bind with the 5 key amino acid residues, but instead it tends to bind with other amino acid residues. With exception for HEGP 17 and 18, they are the two exceptional ones that are known to bind to the binding pocket of the target, where we can see that basically they are known to interact with those tyrosine 36 and arginine 187. Then next, this is one of the examples of the HEGP 01 that I've selected, where we can see from the figure 12, it shows those 3D image for the R configuration, and figure 13 is showing on the S configuration. So this is basically the structure of the HEGP 01 for R configuration, and the bottom one is for the S configuration. So first we'll be focusing on R configuration. what we can see is that when the HAGB01 binds to the binding pocket, what we can see is that basically there is a presence of hydrogen bond interaction. That is, the hydrogen atom of the OH group of the lactone ring of HAGB01 is known to have a significant hydrogen bond interaction with the oxygen atom of the OH group of phenyl ring tyrosine 36 at 1.86 Armstrong. And there is also presence of pi-pi stacking, which can be seen those highlighted in red color, whereby the phenyl ring of the HAGP01 is known to form a very strong pi-pi interaction with the tyrosine 36. But when we see for the S configuration, when the HAGP01, the S configuration binds to a target, there is no pi-pi stacking interaction being seen with the key amino acid residues, but there is presence of hydrogen bond interaction. But this hydrogen bond interaction is found to happen only with asparagine 186 as well as aspartic acid 217, and it does not form a hydrogen bond with the 5 key amino acid residues. And this graph is basically showing the interaction of all the study molecules where it is the HAGP and its benzalidine derivative, both R and S configurations, with the crucial amino acid residues of DNA binding site of P65 target. So first we'll be focusing on coulombic interaction, where the top graph is showing for those R configuration and the bottom one is for the S configuration. When we can see for the R configuration, basically we can see that HAGP07 is known to have a significant coulombic interaction with arginine-187. And those moderate interaction can be seen in HAGP04 with arginine-187. And then HAGP06 also forms a moderate coulombic interaction with tyrosine 36. Those weak coulombic interaction can be seen in HAGP18, where they form the weakest interaction with glutamic acid 39. And for the S configuration, what we can notice is that HAGP11 and 12 is known to form a significant coulombic interaction with arginine 33, while those HAGP03 and HAGP11 and 12, they are known to form a moderate coulombic interaction with arginine-187. And the weakest coulombic interaction can be seen in HAGP11 with glutamic acid 39. So the second form of interaction here is the van der Waals interaction, where we can say that HAGP18 is known to form the significant van der Waals interaction with tyrosine 36. And there is also presence of moderate van der Waals interaction, where you can see that for almost all molecules, they form a moderate interaction with arginine-187. Where for the S configuration, we can clearly see that HAGP14 has a significant van der Waals interaction with arginine-187. Moderate van der Waals interaction can be seen by HAGP14 with tyrosine 36. And the last type of interaction is hydrogen bond interaction, where it can seen clearly for both R and S configuration. Basically, the hydrogen bond interactions are formed with tyrosine 36 residue. And for the R configuration, the significant hydrogen bond interaction can be seen with HAGP01, 02, 06, 08, 09 till HAGP15, where they show the most significant hydrogen bond interaction, while the others shows a moderate hydrogen bond interaction with tyrosine 36. While for the S configuration, only HAGP14 is known to show the most significant hydrogen bond interaction with tyrosine 36. So these graphs is actually showing the calculation of ligand-free energy of binding with the DNA binding site of P65 target using MMGBSA approach. And what we can identify from here is that what we can identify from this graph is basically the R configurations of HAGP and its benzalidine derivatives shows the most stable interaction for almost all molecules as compared to the S configurations, with exception for HAGP 13, 14, and 15, where the S configuration were found to be more stable as compared to the R configuration. So next is the ADME properties for the R configuration. So these are the few parameters that has been selected to test the drug likeness-like property. Basically, for all the parameters being tested, the R configuration of HAGP and its benzalidine derivative that shows a good drug likeness-like property, except for the parameter of predicted aqueous solubility. For HAGP 18 and 19, there is a slight deviation from the accepted range. And similarly, for S configuration, the HAGP 18 and 19 that shows the similar slight deviation from the accepted range for the predicted aqueous solubility. For other than that, all of them shows a good drug likeness-like property. So to conclude, most of the benzylidine derivatives of AGP have shown to bind to the binding pocket of the DNA binding site of P65, which shows that it is possible for these molecules to act as an inhibitor of NF-kappa B. While for the HAGP and its benzylidine derivatives, we see that the S configuration of the HAGP and its benzylidine derivative could not bind at the binding pocket of the DNA binding site of P65 target, with exception of HAGP 17 and 18. While the R configurations of the HAGP and its benzylidine derivatives were found to bind at the binding pocket of the DNA binding site of P65 target. So here we can come to a conclusion saying that the R configuration of HAGP and its benzylidine derivative could be a possible inhibitor of NF-kappa B. And for the dockings, for the ADME prediction, we can see that all the 66 molecules, when they were docked, they show a good drug likeness-like property by obeying the Lipinski rule of 5 and few other parameters, with exception for HAGP 17 and 18 for both R and S configurations, where they show a slight deviation in the predicted aqueous solubility parameter. And this is all my references, and that's all. Thank you.
Okay, Dr. Suresh. Very good presentation, excellent. I can tell you this is an excellent piece of work. It shows a lot of hard work we have done. Docking 66 molecule in a short period of time, it's not an easy job. Definitely, it's a very good work. I would like to ask Dr. Sagi if wish to ask any questions.
Hi, hello Rajkumari.
Yes, sir.
Okay, yeah, good presentation.
Thank you, sir.
And then I appreciate it. Okay, there are very few, very minor comments. When you are introducing the NF-kappa B, you have showed and explained how the mechanism works, how this inhibition works. If not inhibited, how NF-kappa B translocates into nucleus, and then how it gets or how it binds to DNA and then initiate all other cycle of events. But when you are actually docking, I mean, when you are presenting the NF-kappa B mechanism, you have showed the P50, correct or not, subunit?
Can you go to that slide, Rajkumari?
Yes, sir.
When you are introducing NF-kappa B, you explained everything using P50, is it?
No, both heterodimer P50 and P65.
Actually, RELA is the P65.
Oh, that's why I couldn't see. Okay,
No,
okay, I understand.
P65 is also known as RELA. Yeah.
Okay, I understand. I'll take back my comments. The second thing is actually now you are predicting or proposing that some of the compounds are nicely binding to the binding site of P65, isn't it?
Yes.
So if that is the case, is this compound along with the NF-kappa B translocating into the nucleus?
It does not translocate to the nucleus because the target that I've selected is the binding site, the DNA binding site of P65. So it shows that these molecules potentially to act as an inhibitor, meaning that it doesn't enter into the DNA.
No, it doesn't enter nucleus, is it?
Probably it enters because we cannot assure that. What I understand from this work, probably it may enter the nucleus, but even though it enters, it may not bind to DNA since the DNA binding site is blocked by the antagonist.
Yes,
Should be a correct answer, I guess.
Perhaps yes, this might be the possible case. Yes, correct. We cannot say whether we cannot say whether it enters or not, but always possibility is there. It can go through the. I mean, it can translocate into the nucleus.
Do you agree, Rajkumar, with our comments?
Yeah.
And okay, one more third one. Significant van der Waal interactions and all those what we call interactions charts looks nicer, but it will be more nicer if you while you are convincing the audience or the what we call the evaluators, if you highlight where you can see the very significant interaction, maybe using some dotted lines or some highlighting the particular interaction by the particular compound. Correct or not? You agree or not?
Yeah.
You understand my comment?
Yes, sir.
When you are presenting using the bar charts, and then some of the compounds have shown very good interaction, but I do not understand, because you always have to consider the audience have zero knowledge about this what we call modeling or docking studies. You have to assume like that. Then if that is the case, I also myself do not understand, because in the interaction graph has the positive values and negative values, correct or not?
Yes.
So
Would you like to answer, Rajakumari? The how this value significant, like positive value means what, negative value means what. I think that one, if
which is
you
good.
will understand. Yes. Hello, Rajakumari, can you hear?
Yes. Yes, sir.
Would you like to answer?
The more negative the result is, it shows that it is having a more stable interaction, and the positive one is having a weaker interaction.
Because the energy high means unstable interaction, energy less means it is forming stable interaction.
Okay,
from
understood.
From the energy graph, if you see, yep.
If that is the case, for many of the interaction, if my observation is correct, many of the compounds have shown more or less a similar interaction. If you see the pattern.
Correct. Can you go to the graph maybe? Yeah, probably here you can see the Coulombic interaction 7, right? AGP07.
Yes, significant.
Yeah, it shows significance compared to other with amino acid residue arginine 187, which is one of the most crucial amino acid residue for at the binding side. So you can see it is forming strong interaction. This, yeah.
And then van der Waals interaction. Can you please just slide down a bit?
Yeah.
Okay.
Here also the pattern are more or less a bit similar, except one compound is not showing any.
Yeah, except for the AGP, the parent compound, there is almost no interaction with the 5 residues.
Probably because in your derivative, benzylidene derivatives, you have got the benzene ring, because there is a increase in van der Waals interaction, which could be one of the reason.
Okay, understand. Okay, I think. Okay, if you, this is my last question. I'm sorry taking long time. The R&S configurations, if you see the free energy binding, I don't see any significant difference between those two configurations of HAGP, hydro and electrolytes.
There is, when you try to see between like HAGP 06, 02, 01.
I think I should highlight something on this. Yes, first of all, the difference looks a bit less compared to RNS. But if you look at the binding site, S is binding to a different site other than binding. Both are binding, but S is binding to the different site of the receptor, whereas R is binding to the DNA binding site. Can you show that slide, Rajakumari, RNS binding site? Then it will give more significance. So here, if you compare both the binding location where it is binding as well as the binding energy interaction.
Yes.
Yeah. You can see here only the 2 molecules of R isomer is binding at the binding site, whereas most of them are binding at the allosteric site, some other site of the receptor, not to the binding DNA binding site. Whereas in case of R isomer, all of them, I think all of them are binding to the DNA binding site. So in that point of view, you can say like we should not even consider the S binding interaction energy, since it is binding to other side.
Okay, I understand. Okay, that's all. Thank you very much.
Okay, very good presentation. Dr. Thiru, any question?
Yeah. Ms. Rajakumari?
Yes, sir.
It's a very good presentation. Nice.
Thank you, sir.
So I have only 2 questions, ma.
Okay.
So, according to our slide, that P50 and P65 both are entering to the nucleus.
Yes.
I am correct?
Yes.
Why you are choosing 65, not 50?
Because in this case, I'm focusing as for Alzheimer's disease. And there is an article that actually shows that when they do a post-mortem study on Alzheimer's disease patients' brain, they find that the level of P65 is much higher. So it is assumed that P65 is the one that is responsible for the Alzheimer's disease condition. So we want to find molecules that can possibly inhibit the P65.
Okay. Then if you are choosing only your compounds, anthocropolites plus substituted anthocropolites, isn't it?
Yes.
But what is the standard drug? How to compare it with the standard drug? Any drug has actually inhibit that particular site, P65? Any drug which is available in market or any clinical trial drug? Anything is there?
As far as I know, so far there is no any drug that is known to
Mm.
bind to the P65 target.
Mm.
But endographite, there is a report. Sorry. Endographite, there is a report that it binds with the NF-kappa B, P65. That's why I think she has chosen it as a one of the standard molecules.
Okay, this parameter druggability. What are the parameters you are considered as a druggability?
This is the one.
Usually, it's very easy that I wouldn't be able to read it.
Can you make it as a presentation mode?
So it's a rules. I think rule of 5, isn't it?
Yes.
Okay, ma.
Okay, maybe I highlight little bit once again. Actually, 50 properties are measured, out of which only selected are presented here.
Okay.
Like percent oral presentation, rule of 5, solubility, blood-brain barrier.
Okay, ma. Thank you.
Okay,
Thank you,
all the
sir.
best. Very good presentation. Who is going
Thank
to
you,
present?
sir.
Raja?
What? I cannot hear you.
Saja, can you share your screen and present with us?
Okay.
Raja Kumari, you can unshare your screen. We can see your
Oh,
picture.
okay.
Saja, please share your screen. Show your presentation, then you can present.
Is it sharing?
Yeah. All of you click on Saja's block. You can see her presentation. Dr. Sagi, you just click on Saja's block photo. You can see her presentation.
Is it moving?
Yeah, it's moving. You can start.
No. It's moving. Okay, my name is Silda Ahmed. I'm from Master Molecular Medicine. This is my first time to deal with drugs. I hope I will give it a share of proper presentation. So my presentation will be about in silico blocking studies of tocotrienol isomers, alpha, beta, gamma, and delta by targeting three breast cancer receptors, HDFR, HER2, and estrogen. So, What is tocotrienol? Tocotrienol is basically vitamin E. Vitamin E is contains tocotrienol. Tocotrienol isomers have been proven that it. Hello, can you hear me? Tocotrienol isomers are part of the vitamin E, which is basically oil. Tocotrienol is found abundantly in in oil. So tocotrienol has been tried in several studies by different groups. Also, there is a group here in IMU for treating breast cancer. Why for treating breast cancer? They have found that in many cell lines of breast cancer, these tocotrienol derivatives, they can kill the cells or regulate their genes. So to start from the beginning, I will start about the breast cancer. And will develop breast cancer during their lifetime, starting from 30 years old. Second most cause of cancer-related deaths, although there is so many drugs and treatment for this cancer, still it's the second cause of cancer death after lung cancer. Accounts for about 16% of cancer deaths. Human breast cancer, why it's very hard to treat? Because it presents with diverse clinical and histopathological features. So there is no one drug or one target they can attack and treat the cancer, like so many other cancers. But this one has also, because the prevalence is very high and it's affecting women from different ages. So the beginning, gene expression microarray, as I have said, there is a group in our university. They have tried it. Gene expression microarray profiles of breast cancer have demonstrated that tumors can be classified into molecular subtypes. So because they can be identified in molecular subtypes, this group in our university, they have tried to target these genes to treat the cells, and maybe in the future develop a drug to treat cancer or as a prevention. So it can be, the breast cancer can be classified histopathologically and also molecularly into different subtypes. They have different treatment, they have different responses and prognosis. And it's determined by what? By the immunohistochemical strain of estrogen receptor alpha and progesterone receptor and human epidermal growth factor receptor and different other receptors. But the treatments now are targeting these ones. And there's also a type of breast cancer which does not even show all these three categories. So we have 4 tumor subtypes: luminal A, which is ER positive or PR positive, and HER2 and U negative. That means there is no HER2. So luminal B, and I have non-luminal HER2. Also, this one has a positive HER2 and may or may not represent estrogen or progesterone. There is also triple negative, the one I spoke about. This one has no proper targets for these receptors. So this picture will show basically how it's treated. It can be shown here that estrogen, this can be estrogen and progesterone receptors. We have luminal A and luminal B. These are the molecular subtypes here. And they can be treated by chemotherapy or by other radiotherapy or other things. Herceptin, HER2 can be treated by Herceptin or other derivatives of the hormonal therapies or other derivatives of Herceptin or Tenumba. To triple negative breast cancer cells, this is the TNBC. This is HER2 enriched and also may or may not have progesterone and estrogen receptor. Still, if there is progesterone or estrogen receptor, they can be treated. That's why when they are treating this kind of breast cancer cells or breast cancer tumor, they are targeting whether estrogen receptor, progesterone receptor, human epidermal growth factor receptor, and the total the triple negative breast cancer cells, giving combination, no actual treatment for it. So what is tocotrienol? The tocotrienol is vitamin B, E, which exhibit antioxidant properties. It's generic terms that represent derivatives of tocopherol and tocotrienol. Why tocotrienol is more active? Because according to this diagram, which is showing that we have 4 isomers of it. How it is different? It's according to R1, R2, and R3 in the chromonol ring. So this vitamin E, they are far soluble, structure is similar. They have common chromonol ring side chains and C2 at C2 position. Tocopherol has saturated butyl tail, and that's why it's probably not active when cells are treated with it. Breast cancer cells are treated with it, whereas tocotrienol possesses unsaturated isoprene side chain. Depending on the number and position of methyl substitute on the chromonol ring, tocotrienol is classified into different isomers: alpha, beta, gamma, delta. Recent work has shown that tocotrienol can exert direct inhibitive effect on cell growth in human breast cancers with or without estrogen or progesterone receptor. So, my objective will be that to try to see how the tocotrienol can cause cell death. They have found that it could regulate the genes by upregulate or downregulate several genes. And these genes they can downregulate or upregulate are the genes responsible. Some of them are responsible for HER2 and EGFR and estrogen receptor work. So my aim and objective will be to perform visual screening in silico docking and to test the binding affinity of tocotrienol isomers by using crystal structure of EGFR, which is 1M17, and estrogen, which is 2GF9, and HER2 receptors, which is 2GF9, which is 3PPO, respectively. So using what will I do? What did I do? I used Glide version 9.8 Schrodinger 2014 software and used the following steps. As we prepared the ligand, first the tocotrienol, then the protein preparation. I brought EGFR receptor with for an anylquinolizolene inhibitor, which is allantorinab, which is like family of the herceptin, also from the same family, which is a monoclonal, and they bind to its inhibiting site. Then I also got estrogen receptor alpha. Why receptor estrogen? Why I brought the estrogen receptor alpha? Because this is found in abundant amount in the breast cancer cells, more than the other cells. So TGF9, I brought it with tamoxifen because mainly estrogen receptor cell cancer is treated by tamoxifen. And HER2 with kinase domain inhibitor, that one. This one they have proved that by using the crystal structure of the domain kinase inhibitor, by using several co-crystals, they found this is the main inhibiting sites of this receptor or protein. Binding site assessment I did, and then I docked it against the tocotrienol. Then after that I did docking analysis. So this picture will show that at the EGFR level we have this at the binding site, that all the isomers of tocotrienol, alpha, beta, delta, gamma, they are superimposing on the side. And on this picture also we say in HER2 that the tocotrienol isomers, alpha, beta, gamma, delta, they are also superimposing at the site itself. So I'll speak about the receptor itself, why I put superimposing tocotrienol in epidermal growth factor receptors, and why I'm speaking about HER2 and EGFR both, because there is a class called subclass 1 of the receptor tyrosine kinase, superfamily consists of ERBB or epidermal growth factor receptors comprise of 4 membranes, which is EGFR or ERBB1, ERBB2, which is HER2, or an ERBB3, which found that it has its domain kinase is not very effective, and ARBB4. All members have an extracellular ligand binding region, a single membrane spanning region, and a cytoplasmic tyrosine kinase containing the domain. This is the one we are targeting by the tocotrienol. All the isomers tocotrienol were found, as I said before, that they are docking at the same pocket on both receptors. Here is the estrogen receptor. We have two different estrogen receptors. They co-express in many cell types, both composed of 5 domains. Two estrogen receptor subtypes, alpha and beta, are hormone-regulated mediators of the intercellular signaling and gene expression. Regulation of estrogen receptor activity is important in development of homeostasis and treatments of various diseases, including breast cancer. And I think I'm repeating myself. It has been found that an abundant amount in the breast cancer cells, and as shown here, it is still bound to the same bucket where the tamoxifen has been taken from, and they are superimposing on each other. So I'm going to speak after I dock the molecules. We are checking the binding interaction of tocotrienol isomers with EGFR receptor. So I picked the receptors differently: EGFR, herceptin, and estrogen receptor differently. I dock them against the tocotrienol alpha, beta, gamma, delta. So this is with alpha. We found that there is a hydrogen bond, the cyclic oxygenase of chromonol ring interacts with amino acid group of glycine here, and similarly the hydrogen atom of chromonol ring interacts with oxygen atom carbonyl group in leucine 768. On the other side, I found also beta. There is bistacking. It could be acceptable bistacking interaction, and the hydrogen bond also is found between the chromonol ring interacts with the oxygen of the carbonyl group of ASN 0.818, and with significant bonding, which is 1.86. Delta tocotrienol, there is a hydrogen bond in between the oxygen atom of carbonyl group leucine 7681 and the hydroxide of carbonyl ring, cyclic carbonyl ring. Also, in gamma tocotrienol, there is also bistacking ring, and it's also there is a significant hydrogen bond in between the cyclic hydroxide and the oxygen atom of the carbonyl group of ASN 818, and it's a significant hydrogen bond. Van der Waals and the Coulomb negative, that means showing that there is a strong interaction between this between the receptors and the tocotrienol. Also, I docked the tocotrienol against the isomers of estrogen receptors. Also, there is a strong hydrogen bond in alpha tocotrienol in between the chromosomal ring interacts with oxygen atom of the amine group of 383. Tryptophan amine group. I'm stuck. Is it coffee?
Hello. What happened? Hello, Saja.
Hello. I don't know. I'm blank.
Why you are
yes,
blank?
I'm blank. Everything is blank in front of me.
Nothing
I
is
don't
blank.
know.
You just see your
Everything.
slide. We can see your slide. Don't worry. You just present your slide. Good.
Okay. So here is the binding interaction of tocotrienol isomers with estrogen receptor. This one is ADFR. With estrogen receptor, we can see also the cyclic oxygen of chrominol ring is binding with aspartate 351, and also in beta tocotrienol, the hydrogen bond also is found making a significant hydrogen bond in between the hydroxide group of the cyclic oxygen and the carbonyl group of tryptophan 3H3. In delta tocotrienol, we can find a pyrostacking, and gamma tocotrienol we can find also a pyrostacking. In what is the significance of these? In real studies, when they did it in vivo and in cell lines, they found that gamma tocotrienol and delta tocotrienol, they may exhibit more effect on killing or decreasing the proliferation of breast cancer cells of different types. And here they are also showing more bypass-stacking bond and more hydrogen bond. Also in gamma tocotrienol, we found also there is a hydrogen bond between the carbonyl ring, which interacts with the oxygen atom of carbonyl group of leucine 3H7. Also there is a bypass-stacking ring, bypass-stacking interaction between phenyl ring of carbonyl ring, which interacts with phenyl ring of phenylfluorophore. Okay, this is also the van der Waals. I'm showing van der Waals in blue, Coulombic in red, and the hydrogen bond in green. Also, we can find there is bonds, but still, regardless of that, still in delta, I don't have a strong hydrogen bond. Here also the binding of tocotrienol isomers with human endothelial growth factor receptor. This one also we can find there is a strong bond in alpha tocotrienol between the aspartate and the cyclic and the carbonyl group of between the aspartate and the hydroxide group of the cyclic group of carbonyl ring. And the hydrogen atom of carbonyl ring interacts also with oxygen atom of carbonyl group of aspartate 808, and it's also significant bond. In delta, there is also strong hydrogen bond can be found in between the aspartate and the carbonyl ring. Also here between the aspartate and the carbonyl ring, we can see at the same site. Van der Waals and Coulombic of all the receptors, alpha, beta, gamma, delta. They are almost the same, except here delta also showing not a good van der Waals interaction. Here is the calculation of the free energy of EGFR and HER2 and ER. I put them together to compare, and as we can see, they all have they are on the negative side. That means that they show a good free energy. But still, it's almost the same by using the Schrödinger program. Here's the prediction of the ADME of this of tocotrienol isomers alpha, beta, gamma, and delta. We can see that they are safe and they are not causing toxicity, and their solvent accessible service there is more than 500. Their donor HCP is 1. Human absorption, oral absorption is 1. This is acceptable. So in conclusion, I will say that all the tocotrienol isomers alpha, beta, gamma, delta have exhibited good affinity to the binding pocket of EGFR, estrogen receptor, and HER2 receptor, especially delta and gamma, mostly in estrogen and in EGFR. All the tocotrienol isomers showed good drug likeness properties, obeying the Minsky rule of 5. So maybe, therefore, tocotrienol isomers could have a promising therapeutic or preventive role in breast cancer. This is my references. Thank you.
Okay, good presentation. Dr. Sagi, any question?
Hello, Shahjam.
Yes.
Okay, yeah, good presentation, very impressive, thank you. Okay, before I go to the conclusion set, because I have only 2 questions to ask you.
Okay.
One is actually these tocopherols or tocotrienols, I don't know, I think which.
Tocotrienols.
Tocotrienols.
Yeah.
Very, very established molecules, isn't it? A lot of research has been done and still going on. So the work you have presented should have somewhere already in vitro or molecular studies should have been carried out. So maybe did you find any literature to compare your predictions with the actual findings in the experimental lab? Any correlation with your findings with the actual molecular studies or whatever in vitro studies?
Actually, molecular studies, they found that the IC is about 7 in
Okay, sorry to interrupt. Can you unshare your screen so that you can see you while talking to you? Raja,
okay.
just unshare it.
How will I unshare it?
Just click the share button again. It will
Okay.
unshare. Okay, you can see you now.
Okay.
You can answer now.
Yes, there is a lot of research has been done in cell lines, and they have found that the IC of tocotrienol is about 7. It's about 7, but they have not proven anything in real life, because according to my professor and according to the study, when they have applied the tocotrienol, it has one problem, that because it has this, it can change, and it has this, it easily oxidizes when taken orally. So this is one problem. And second problem, when they did the experiment in vivo, I mean in human, in vivo it's also showing good in animals in the labs. In humans, when they predicted its value, they did predict it with the mortality rate. They did not predict it with decrease of the size of the cancer or what happened to the cell lines. They did not check the cancer itself, what happened to it, only by the. So now they are repeating the trial one more time. Did I answer your question or no?
Okay, maybe I will talk to you.
You can ask me again, no problem.
Yeah, you explained to you, but maybe I'll talk to you when actually, because through online your voice is not very clear. Maybe sometimes you got to talk very slowly, pause sometimes, like that.
Okay, I would love to talk to you.
Okay, no problem. And then one more question.
Okay.
Sorry. When you are if you go to your conclusion, you don't need to go to the slide, just to remember, you have concluded your study saying that almost all tocotrienols have shown very good binding properties or something. So if that is the case, did you, is there any take-home message or a very significant discovery from your study? Actually, narrowing down which isomer or which one to further to, which one to take into further studies or something to take as a lead. You are telling almost all are showing good one.
Yes, really, in virtual studies, they are almost showing the same, but the bye-bye stacking is only formed mostly in delta and gamma, which proves, like in vivo, this is more say it should be targeted if they are going to use it as a drug or prevention for breast cancer.
Okay.
Because it really regulates in triple-negative cancer cells. The one does not have any treatment. It regulates 46 genes, but still they don't know, because online, if you check, there is no docking of tocotrienol against breast cancer and how they are going to use it as a treatment. No docking has been done, so they don't know how to make it as a drug. Not yet. It's not given as a drug at all. It's given as a supplement only.
Okay. Okay, understand. Thank you. That's all from me.
Dr. Thiru, would you like to ask any question?
Yes, so presentation is very good. Your presentation is very nice. Can you hear me?
Thank you. Yes.
Actually, this tocotrienol is already established, a well-known molecule, isn't it? Tocotrienol, isn't it? I am not asking about from your presentation. Generally, I am telling something. Tocotrienol main problem is only bioavailability, isn't it?
Yes.
Can you do this drug as a directly, can as a implant and all is possible? Because anyhow you are going to do for breast cancer, isn't it?
Yes.
We can give it as an implant to kill the cancer cells.
I don't know. That's why we docked it. Nobody has docked it against the receptors at all. Even I have a master student, I think it's not published. He did dock it against the genes, but never against the receptors, because it's really very hard to find the receptors with but although they are in the data protein base, but you have to validate them.
Okay, wish you all the best. Then you have to publish the paper, isn't it?
Thank you.
Okay.
Thank you.
Okay, good luck. Saja, you can mute your mic. Now I think we will start with Farida. You can start your presentation and try to finish it as quickly as possible, since today is Friday and everyone is waiting to close the door, So try to finish it early. Farida.
Right.
Share your screen. Left hand side you can see. Farida. Just share. Yeah. Share your screen. From the left hand side of the top you can see the green color button, share screen. You share that, then only you can see your presentation. Slides. She got stuck in her own computer. You should have been ready by this time.
No, it's hanging. That is why I close it. I'm going to use mine. It's not okay. Can you? Can you see it now? Is this? Okay. Can you see it now? Yes. Okay.
Yes, we can see your slides.
Can I start now?
Yes, start.
Hello, sir.
Hello.
Okay, sir. Okay, good evening, everyone. Hello? Hello, sir.
Hello, we can hear you. We will not be talking to you. Just present.
Okay.
All the best.
Okay, good evening, sir. Thank you, sir. Good evening, sir. I'm here to give a seminar on in silico docking studies of some marketed selective COS2 inhibitors and novel pyrimidine derivatives. This is the outline of my seminar. Introduction, I'm going to give a brief introduction on cyclooxygenase, mechanism of inflammation, objectives of my studies, docking study protocols, prediction of ADMET properties, the result of the study, conclusion and references. The introduction. Cyclooxygenase is a homodimer enzyme that catalyzes biosynthesis involving conversion of arachidonic acid into prostaglandins. Generation of prostaglandins are one of the most important factors in inflammatory processes. Therefore, cyclooxygenase is a target of non-steroidal anti-inflammatory drugs. Cyclooxygenase exists in two isoforms, the constitutive cyclooxygenase 1 and the inducible cyclooxygenase 2. Long-time use of this non-steroidal anti-inflammatory drug is associated with adverse side effects such as gastrointestinal ulcer and renal function suppression. This side effect associated with non-steroidal anti-inflammatory drugs are due to inhibition of cyclooxygenase 1 pathway. Therefore, selective cyclooxygenase 2 inhibitor associated with an increased risk of cardiovascular diseases. Hence, design of new compounds with good binding affinity and satisfactory ADMET properties that will selectively inhibit COX-2 is of interest. This mechanism of inflammation, as we can see, cyclooxygenase is responsible for synthesis of prostaglandins that maintain our physiological functions, while cyclooxygenase 2 is responsible for synthesis of prostaglandin that induces inflammation, that is pro-inflammatory prostaglandin. These are the objective of my study: to perform in silico docking studies of indomethacin, Silico and novel pyrimidine compounds with crystal structure of cyclooxygenase 1 and cyclooxygenase 2 isoenzymes to predict binding affinity as well as selectivity of novel pyrimidine compounds. And thirdly, to determine ADMET properties of novel pyrimidine compounds. The protocol, that is docking study protocols. First and foremost, the ligands were prepared using lead prep module of Schrödinger. The protein preparation involved pre-process of protein, that is addition of hydrogen to the raw protein downloaded from PDB, that is protein database, then minimization of the protein, optimization of the unprocessed cyclooxygenase 1 and cyclooxygenase 2 isoenzymes. The thirdly, grid generation. Grid generation involves assigning active site to the target, that is assigning active site to the both cyclooxygenase 1 and cyclooxygenase 2. The fourth is docking. Then the docking were performed using Schrödinger. Indomethacin novel pyrimidine derivatives were docked with crystal structure of cyclooxygenase 1 and cyclooxygenase 2 isoenzyme with PDB ID, that is the protein database ID of 3N8S and 3LN1 respectively. While the ADMET properties were to be predicted using kick-prop module of Schrödinger. Results and discussion. Though in this discussion, I didn't really give the 3D view of the indomethacin and the Silico because it has been established that they are inhibitor of both cyclooxygenase 1 and cyclooxygenase 2. Okay, this is the result of my discussion. That this is the outcome of my study. This slide shows the key amino acids responsible for binding of ligand in cyclooxygenase 1 binding site. As you can see, leucine 352, serine 353 are the key amino acid responsible for the binding of ligand at the binding site of cyclooxygenase 1. Okay, this 3D view shows the superimposition of all my docked molecules. As we can see, all the docked molecules bind at the same binding pocket of the binding site of the target. None of the molecule falls outside the binding site. All of them bind at the same at the binding pocket of the target. This slide shows our pyramid 3D view of our pyramid novel structures. Our pyramid structures right now phenyl ring of 355 forms significant pi-pi stacking interaction with benzene ring of the noble compounds. Noble compound. Why also the carbonate group of the noble compound also form a significant interaction with hydrogen bond interaction with amino group of arginine 120. This is the binding site, that is the binding site of cyclooxygenase 2 isoenzyme. These are the amino acids responsible, that is the key amino acids that are responsible for the binding of ligands at the active site of cyclooxygenase 2. Likewise, this is the superimposition of all the molecules at the active site, that is at the binding site of cyclooxygenase 2. Likewise, same as the cyclooxygenase 1, all the molecules, all the dock molecules fall within the same range, that is they bind at the active site of the cyclooxygenase 2 receptor. Likewise, this is another molecule showing interaction, that is the our novel pyrimidine derivative amino group binding, having a good hydrogen bond interaction with OH group of serum 516, and likewise the methoxy group of novel pyrimidine derivatives showing interaction with OH attached to phenyl ring of tyrosine 341 and amino group of arginine 106. Wow, yes. This is the van der Waals interaction of the studied compound with the crucial amino acid residue at the active site of cyclooxygenase 1. As we can see, most of the molecules have good stable interaction with the key amino acid residue at the COS2 binding site. This is the electrostatic or the Coulombic interaction of all the study compound with the crucial amino acid residue at the active site of COS2. Not all most of the molecule didn't form a kind of a stable interaction with the key amino acid. This is the third graph. This third graph shows the van der Waals interaction of all the study compounds with crucial amino acid residue at the active site of cyclooxygenase 2 isoenzyme. Virtually all the molecules, aside from three molecules, aside from three compounds, virtually all the molecules shows a very, very stable interaction. That is a good interaction with the key amino acid at the key amino acid residue at the active site of cyclooxygenase 2 isoenzyme. This graph shows electrostatic interaction of the study compound with crucial amino acid residue at the active site of cyclooxygenase 2. Likewise, same thing like cyclooxygenase 1. Most of the compound didn't really show a kind of a stable interaction, but those that show stable interaction, they show a very, very significant stable interaction. For example, sample structure 13 shows a very, very stable interaction at the with the crucial amino acid residue at the active site of cyclooxygenase 2 isoenzyme. This is the this graph shows calculation of ligand free energy of binding with cyclooxygenase 1, binding of ligand with cyclooxygenase 1 using MMM-GBSA approach. As you can see, virtually all of them show 18 that falls within the positive range. That is, it shows a kind of unstable interaction with the. It shows an unstable interaction with the receptor. Likewise, this the sixth graph, which shows the binding, the ligand binding, ligand free energy of binding with COS2 using MMGBSA approach also. In this graph, all the molecules shows stable interaction. Stable interaction with the most structure 15 shows a very, very stable interaction, while structure 7 shows moderate interaction. Based on the free energy, free binding energy, it can be deduced that our novel pyrimidine derivatives are well anchored at the binding pocket of cyclooxygenase 2 isoenzyme compared to cyclooxygenase 1 binding site. This table shows the prediction of IDME properties. most of all the pyrimidine derivatives, that is all the novel compounds, shows a very, at least, good to moderate ADME property, that is drug-like property, ranging from the human oral absorption. All of them show a very, very good human oral absorption. In fact, all of them, actually, based on this ADME, all the novel compounds show a very, very good ADME property. In conclusion, the design of pyrimidine derivatives does not only show more stable interaction with COX-2 compared to COX-1 isoenzyme, but also have satisfactory ADME and good drug-like properties. Hence, this implies that our novel compound may act as selective COX-2 inhibitor. These are my references. Thank you very much.
Okay. Any question, Dr. Sagi? It was a good presentation. Dr. Sagi, would you like to ask any questions?
Actually, in fact, I do not have any questions. The presentation is very nice and straightforward, and I can see because COX-1, COX-2 are also very established targets, and then the presented compounds also docked very precisely, and everything nice. At the moment, I'm sorry, I do not have any questions. Please proceed.
Okay, like Dr. Thiru. Farida, can you unshare your screen? We can see you.
Okay, sir.
No.
Dr. Thiru would like to ask a question.
It's a very nice presentation.
Thank you, sir.
You are using COX-1 and COX-2 both receptors, isn't it?
Yes, sir.
But slide number 10, you told me that COX-1 is very good interaction is there, isn't it? Slide number 10 actually.
Okay.
But actually in the introduction you told me that COX-1 is for the responsible for side effects.
Yes, sir, because cyclooxygenase blood inhibition. Okay, sir. Can I go ahead?
Yes.
Okay, actually, inhibition of cyclooxygenase 1 is very, very catastrophic in the sense that it inhibits production of prostaglandin that are responsible for the maintenance of our normal physiological functions. So we don't want to synthesize a compound that will inhibit cyclooxygenase 1. Rather, we want a compound that will selectively inhibit cyclooxygenase 2.
But you showed me that one slide, slide number 10, that also very good interaction with COX-1.
Yes, that is our novel compound. That is the outcome of our study.
How come the COX-1 also showing good interaction?
So then how come you selectivity, how do you know, how can you say this is selective to COX-2?
Yes, it's selective COX-2 because the overall GB free energy binding shows that COX-2, that is the compound shows more binding affinity towards COX-2 than COX-1.
That means it is not 100% selective. It is partially selective. It probably would be 75% to 80% selective, but it is still have the potentiality to bind with the COX-1. So the compound that you have designed, it's not actually 100% COX-2 selective, but it is partially, it has more affinity towards COX-2 compared to COX-1. So maybe you need to screen some more compound to check to improve, the binding affinity towards COX-2 compared to COX-1.
Okay.
Sorry, you didn't show any compounds. Is any? You didn't show any compounds. Pyrimidine derivatives is it confidential?
Because I didn't mention it's a new compound, sir.
That is why it's a confidential.
No, you can show your dock. Some of the molecule you have docked, and right, you can show the
this is the dock.
That's yeah, the slide you show the some pyrimidine compound that you have docked.
Okay.
Can you share your slide and show?
Okay, sir.
And just she should have included all the list of. I also last moment did not check. Can you share your slides?
Yes, sir. I will
need to share.
Okay. I think it's not shared.
You share. You click the share button.
It's okay. It's okay. It's okay.
Okay.
Yeah.
This is the docked molecule design compound.
But I couldn't able to understand what is the structure of the molecule.
Oh, it's a pyrimidine derivative, sir.
You should have drawn the structure, scaffold, like how others have shown.
Okay.
Like how Rajakumari have shown. The structure, the derivatives, like that, you should have shown. Which derivative it is. Because from 3D, sometimes it is difficult. Or you would have shown the structure here on the right hand side. How the structure, which part is interacting.
Okay, sir.
At least you should have maintained the 20 or how many compound you docked?
21.
At least you should have shown, which are the compounds you screened. Unfortunately, I also did not check. Otherwise you can see the Rajakumari, she has shown nicely all the 64 molecules and the derivatives. Anyway, this is just for next time, because when a unknown person see, they very difficult for them to understand what is the scaffold, what are the substituent and derivative, right? So next time take care. Anyway, presentation was good. All the best. So we stop our presentation here. Wish you all the best. Okay. Good luck. See you next time.
Happy